Hi, we'll start our lecture about tissue culture by a brief introduction and historical background. Tissue culture was first devised at the beginning of the 20th century to study the behavior of animal cells in non-in vivo setting. The technique was done first using cells migrated from fragments of tissue. This segregation and plating out of cells was first demonstrated by Rowe 1916. In 1948, masking fibroblast was the first cloned cell line. In 1950, trypsin, a protease, was used to subculture as described by Delbecco to obtain monolayer culture and the generation of a single cell suspension by transtrypsinization. In 1952, Gay established the first continuous human cell line, HeLa, which is a human epithelial cervical cancer, which was subsequently cloned by Puck in 1955. The introduction of antibiotics facilitated the long-term cell line propagation. In 1907, Harrison chose a frog as his source of tissue because it was a cold-blooded animal, and tissue origination is more common in lower vertebrates. Embryonated hen's egg was the favorite choice because the ease of the access of the different tissue, and as well it is a warm-blooded animal. Rodent brought mammals to the forefront as a favorite material, and it had the advantage of producing continuous cell lines. Here from this bar chart, you can see the number of hits of articles and published paper in PubMed for cell culture starting from 1965. And you can see that the number of hits is increasing over years. So tissue culture is a term to include organ culture and cell culture, while organ culture implies a 3D culture of tissue retaining some or all of the histological feature of the tissue in vivo. Cell culture refers to a culture derived from dispersed cells taken from original tissue, from a primary culture, or from a cell line by enzymatic, mechanical, or chemical disaggregation. There is a lot of advantage of tissue culture, including the physicochemical environment. You can control pH, temperature, osmolality, and dissolved gases, as well as physiological condition like controlling of amount of hormone and the nutrient concentrations. As well, you can control the microenvironments, including the matrix, cell-cell interaction, and gaseous diffusion. You can have the availability of selective media for cell cloning and have a cell line that is homogeneous. As well, you can perform any cytology, DNA profiling, or immunostaining for characterization of a cell line. You can do as well stock storing for the cell lines produced in cell culture in liquid nitrogen in the process of preservation. You can do as well validation and accreditation and to identify the origin, the history, and the purity and the authenticated and recorded cell line. You can do replica and the variability, so you can easy quantitation and minimal statistical analysis. Tissue culture as well can do reagent saving because there are reduced volumes are used and there is a direct access to the cell, so that's lower the cost. And you can as well have the ability to define the dose and the concentration and time and the control of the concentration and time. There is availability as well of micro-titration and robotics, that's a mechanization. You can scale up, a number of replica can be increased substantially. It's a time saving using cell culture or tissue culture because you can do a lot of replica, and as well the assay time is reduced in order of magnitude. There is a reduction, of course, of animal use for the you can do cell culture for the cytotoxicity and the screening of pharmaceutics, cosmetics, and etc. And these are the some of the advantage of tissue culture. But there are some limitation for the use of the tissue culture. First thing is the necessary expertise. So scientists need to work on the cell culture. They need to understand how to do sterile handling, how to avoid chemical contamination, how to detect microbial contamination, and be aware for detection and misidentification. Environmental control. So, a tight control on the environment around the cell culture of isolation and the cleanliness of the workplace, incubation and pH control, and you need to know how to dispose biohazards. Quantity and cost. The capital equipment for scaling up media, serum, and disposable plastic is a bit expensive. Genetic instability and the phenotypic instability. Cell culture may behave unstable with respect to heterogeneity and variability and de-differentiation and adaptation, and might be selective over growth. Identification of cell type. Markers are not always expressed in cell culture, and histology difficult to recreate and atypical. The geometry and the microenvironment changes cytology. So, what are the types of tissue culture? There are three main methods of initiating culture. Organ culture, the architecture characteristic of tissue in vivo, is retained in culture. The tissue is cultured at a liquid gas interface, grid or a gel, which favors the retention of a spherical or a three-dimensional shape. While the primary explant culture, it is a fragment of tissue, is placed at a glass or on the glass or a plastic liquids interface, where after attachment there is a migration is promoted in the plane of the solid substrate. While the cell culture, a tissue or outgrowth from the primary explant is dispersed mechanically or enzymatically into cell suspension, which can be cultured as an adherent monolayer on a solid substrate or as a suspension in culture media. The formation of a cell line from primary culture implies an increase in the total number of cells over several generations, and the dominance of the cells with the capacity for high growth, resulting in a degree of uniformity in the cell population that give rise to specific cell line. So here you can see how it looks like in case of organ culture. There is a tissue at the gas liquid interface, and the histological structure is maintained. The explant culture, you can see that tissue at a solid liquid interface and cells, as shown by the arrows, migrate to form outgrowths. The dissociated cell culture, you can see that the disaggregated tissue cells form a monolayer at solid liquid interface. While the organotypic culture, there are different cells co-cultured with or without a matrix, and the organotypic structure is recreated. In organotypic culture, presence of more than one cell type interacting as they might in organ of origin. This system is biochemically and physiologically more similar to the in vivo setup. The biology of culture cells. So the validity of a culture cell as a model of physiological function in vivo has frequently been criticized. Cells do not express the correct in vivo phenotype because cells' microenvironments have been changed. Many of the hormonal and nutritional stimuli are absent. This creates an environment that favors the spreading and migration rather than the expression of differentiated function. So the influence of the environment on the culture is expressed via five routes: the nature of the substrate on or in which the cell grow, the degree of contact with other cells, the physicochemical and physiological constituents of the media, the constituents of the gas phase, and the incubation temperature. Cell adhesion. After a tissue disaggregation, cells need to attach and spread out on the substrate before they will start to proliferate. It was found that cells would attach spread on a glass that have a slightly negative charge. Cells will also attach to some plastics, polystyrene, if treated with high energy ionizing radiation. Substrates pre-treated with fibronectin or collagen or derivatives such as gelatin will help cells to attach and proliferate. Cell adhesion is mediated by specific cell surface receptor for molecules in the extracellular matrix. So it seems likely that spreading may be preceded by the secretion of extracellular matrix protein and the proteoglycans by the cells. What's the cell adhesion molecules? There are three major classes of transmembrane protein have been shown to be involved in cell-cell and cell substrate adhesion. The first one we call it CAMs. It's either calcium, CAMs is calcium independent and cadherin is calcium dependent. Both of them are involved primarily in an interaction between homologous cells. The second class of adhesion molecules is integrins, and these integrins mediate cell substrate interactions. Receptors for matrix molecules such as fibronectin, laminin, and collagen bind to them via a specific motif, usually containing the three amino acid arginine glycine aspartic acid RGD sequence. The third class of adhesion molecules is transmembrane proteoglycans, also interacting with the matrix constituents such as other proteoglycan or collagen, but not via the RGD motif. The proteases can digest some of the extracellular matrix and may degrade some of the extracellular domains of transmembrane protein, allowing attached monolayers cell culture to become dissociated from each other. So this figure illustrates the types of cell interaction between cell-cell interaction and cell matrix interaction. So you can see a monolayer of cell. This monolayer sits on a basement membrane, and there is a connective tissue below it termed the stroma. Each cell can have molecules that bind the cell to each neighbor cells and molecules that bind the cell to the matrix. So here you can see the first, there is some integrins. These integrins, as enlarged by the figure, they are receptors with intracellular signaling domain and variable extracellular domain bind to the RGD motif in fibronectin or laminin or collagen. So you can see as well, proteoglycans is a low affinity that doesn't depend on RGD domain. And you can see that this proteoglycan, this transmembrane receptor without signaling domain bind matrix of proteoglycans, collagen, and growth factors. You can see as well the cadherins. The cadherins uses calcium, which facilitate the binding between neighboring cells. These cadherins are attached internally inside the cells by intermediate filaments in the desmosomes. And cadherins as well can exist in the adherence junction. And of course, actins shares a lot in the cytoskeleton that aid in the process of interaction between cells and the matrix. So if we look closely about the differentiation from the normal in vivo differentiation from stem cells, you can see that in vivo, the pluripotent stem cells start to do a lineage, is differentiated into a lineage stem cells within 36 hours, so to maintain a stem cell pool. Soon after that, there is another differentiation to form a committed progenitor cells within 18 hours in a process of amplification that soon differentiated to a non-proliferating differentiating cells that soon give rise to the terminally differentiated cells with regulation and adaptation of the amount of these cells. Of course, there is a process of attenuation for the committed progenitor cells as a mode of regulation, and this normally what happens in vivo for stem cells. So if you grow these stem cells in culture, there is a blocked differentiation you can see. So normally they take the first step of lineage stem cell in a longer period, 72 hours, and skip two processes of committed progenitor and non-proliferating differentiated, but still go in a process of amplification, and the attenuation will be less pronounced until it reaches to a differentiation state which is limited by the need to proliferate. So you can see that all of the cells have been terminally differentiated. So here in this slide we show how the stem cell is differentiated in case normally in vivo and in case in vitro in cell culture. So initiation of the cell culture. Briefly, a culture is derived either by outgrowths of migrating cells from a fragment of tissue or by enzymatic or mechanical dispersal of the tissue. The primary culture is the first in series of selective processes that may in the end give rise to a relatively uniform cell line. In primary explantation, only cells that survive the disaggregation process can and adhere to the substrate or survive in suspension will form the basis of the primary culture. Cells that are capable of proliferation will increase. Some cell type will survive but not increase, and yet others will be unable to survive under a particular condition of the culture. After confluence is reached, cells whose growth is sensitive to contact inhibition and density limitation of cell proliferation will stop dividing, while any transformed cell which are insensitive to density limitation will tend to overgrow. Keeping the cell density low helps to preserve the normal phenotype in culture. The evolution of cell lines. After the first subculturing passage, the primary culture becomes known as a cell line and may be propagated and subcultured several times. With each successive subculture, the component of the population with the ability to proliferate most rapidly will gradually predominate, and the non-proliferating or slowly proliferating cells will soon be died and diluted out. In the presence of serum and without a specific selective condition or selection media, mesenchymal cells derived from the connective tissue fibroblast or vascular elements frequently overgrow the culture. All these steps have this has given rise to some of the very useful cell lines, including WI38 human embryonic lung fibroblast, BHK21 baby hamster kidney fibroblast, L cell, which is a mouse subcutaneous fibroblast. The normal cell can divide a limited number of times. Hence, cell lines derived from a normal tissue will die out after a fixed number of population doubling. This is a genetically determined event involving several genes and is known as senescence due to the inability of the terminal sequence of the DNA in the telomeres to replicate at each cell division. The result is a progressive shortening of the telomeres until finally the cells is unable to divide further. Exception to these rules are the germ cells and the stem cells, which often express the enzymatic telomerase, which is capable of replicating the terminal sequence of DNA in the telomere and extending the lifespan of these cells. In tumor cells, cells are said to be immortal due to the transformation, which implies an additional alteration in gross characteristics like anchorage independence, loss of contact inhibition, and loss of contact inhibition and density limitation of the growth that will often but not necessarily correlate with tumorigenicity. This figure illustrates clearly how the evolution of cell line occurs. So you can see here on the x-axis the weeks in culture, and on the y-axis is a cumulative cell number. You can see initially in the first up till the first 2 weeks cells are termed primary culture. Then you do a subculturing denoted here by first subculturing, second subculturing, like every 1 week at a time. So the population of the cells is increasing, and in this condition, from week 2 to week 14, as an average, is a serial passaging or serial passaging or subculturing. And these the cells here have a finite cell line. After week 4, if it's due to some condition like mutation or immortalization, the cell can be transformed. And in this case, we term it a continuous cell line and can be propagated indefinitely. This figure shows the difference between the frequency or chromosome numbers of a normal human glial cells, which is a finite cell line. This cell line is not transformed or immortalized. And the chromosomal number of a continuous cell line from human metastatic melanomas. So you can notice the frequency of chromosomes is much higher in the metastatic melanoma cell line. So what is the requirement of a cell culture? You need to know the number of users, how many people will work in the facility, how long will they work each week, what kind of culture will they perform, the space, what space is required. And normally it's a 4 to 2 to 1 to 1. So you need to know the culture operation area, the area that will need to put in it the laminar flow hood, the cell counters, centrifuge, and a lot of other equipments and reagents. The wash up and the preparation and the sterilization area, the storage area, the incubation area. You need to know where to put is the location of preparation area. The facilities for washing up and for sterilization should be located close to the aseptic area on an outside wall to allow for heat extraction from autoclaves. As well the storage. What is the scale of the work expected? How much storage space will be required for disposal? What requirement for storage in liquid nitrogen? The access. The doorways are wide and high enough. The ceiling have sufficient clearance to allow the installation of equipment. The access space for maintenance of equipment as well ensure that the tissue culture is accessible to animal facility if users require to access the animal facility. So you need a space for double change of lab coats. Quarantine, contaminant, and sterility. Separate room as a quarantine or a contaminant room. Separate aseptic area room must be separated by a door or airlock. These questions will enable you to decide what size of facility you require and what type of accommodation. So here we will see is a design for a small lab culture, for a small tissue culture lab that suits for a 2 or 3 person max. So you can see there is a place for an air intake or location for air conditioner. There is a regulator incubator. There is a humid CO2 incubator, carbon dioxide supply, refrigerator, freezer, bench with shelf over cryo freezer and deer under, liquid CO2 cylinders with automatic changeover, main entry is double asymmetrical door, laminar flow hood, usually it's a class 2 biological safety cabinet with a total extract, service cart, storage rack, sterilizing oven, autoclave, air extract, oven, washing sinks and soaking sinks, inverted microscope for observing cells, cell counter, water purifier and sink, centrifuge, preparation bench and water bath. And that's a design for a small tissue culture lab. Of course, you need a deionized water. For a medium-sized tissue culture lab that suits a 5 to 6 person, most of the stuff are still present, but in large scale, so you need a two-tiered regulator, two-tiered humid carbon dioxide incubator, a carbon dioxide supply, two-tiered humid carbon dioxide incubator again, liquid carbon dioxide cylinder, the main entry as well, and service cart. You have as well a reagent shelf for the steriling fluid. The air intake is high level air intake, refrigerator, and freezer, as well as a centrifuge. Here you can see the design for medium-sized tissue culture lab with adjacent preparatory area. And you can see that the preparation area is separated from the cell culture lab with all the equipment need for sterilization and preparation of chemicals. These designed for a large tissue culture lab that suits around a maximum up to 30 person, and you can see that the space is huge compared to the other designs, and most of the equipments are repeated, and you can see that there is almost like 5, 6, 7, 8, 9 laminar flow for working with the cell culture. Storage. So storage must be provided for the following items to ensure that the sterile and non-sterile are kept separate and clearly labeled. So storage must be provided for sterile liquids at room temperature, liquids like media at 4 degrees centigrade, serum trypsin, glutamine, etc. at minus 20 and minus 70. As well for sterile and non-sterile glassware, including media bottles and pipettes. Sterile disposable plastics that is used in the cell culture. Screw caps, stoppers, and other such sterile and non-sterile items. Apparatus such as filters, sterile and non-sterile. Gloves, plastic bags, and other disposable items. Liquid nitrogen to refill freezers. Liquid nitrogen should be stored in two ways. In dewars that take up till 50 liter under the bench, or in large storage vessel up to 150 liter on a trolley. A pressurized tank for carbon dioxide that is refilled regularly. A space for storage cylinder for carbon dioxide. So, what is the risk of contamination in cell culture? Contamination by microorganisms is a major problem in tissue culture. Bacteria, microorganisms, yeast, and fungi spores may be introduced via the operator, the atmosphere, work surface, solution, and many other sources. Aseptic technique aims to exclude contamination by establishing a strict code of practice, ensuring that everyone use the facility adhere to this code. Catastrophes can be minimized if culture are checked regularly by eye and on-face contrast microscope. Culture are maintained without antibiotics. Reagents are checked for sterility before use. Bottles of media or other reagent are not shared with other people or used for different cell lines. The standard of sterile techniques is kept high at all times. What's mycoplasmal infection? Mycoplasma is invisible under regular microscope. It's a major threat. It is essential to back up visual checks with mycoplasma test, particularly if cells growth appears abnormal. You can see here you can see here the layout of a tissue culture area has changed over years. In the top figure, you can see how ages ago they were working by cell culture, and then they moved to work over a bench and then inside a hood. Recently, the image will look different. So here you can see what is the difference between the laminar flow hood laid out correctly, and the laminar flow hood being used incorrectly. In the bottom figure, you can see that the hood is too full, and many items obstruct the air intake at the front, destroying the laminar airflow and compromising both contaminant and sterility. And you can see here today's laminar flow hood, how it's organized and how it's big. Another picture to today's laminar hood. Thank you.